Welcome to Bioratna. Dear students, I am Chandana Sengupta and our today's topic of discussion is Lac Operon from the chapter Molecular Basis of Inheritance. This lac, it stands for lactose. What is lactose? Lactose, it is a disaccharide. It is a disaccharide made up of two monosaccharide units. One is glucose and another is galactose. So lactose is a disaccharide. We leave it at that and now we see what is operon. This operon concept, this was elucidated, this was cleared, this was given by the geneticist Francis Jacob and the biochemist Jacques Monod. What is operon? Operon is a polycistronic gene. A polycistronic gene. What is a polycistronic gene? There are many structural genes which are regulated by a common promoter. So, a polycistronic gene regulated by a common promoter and of course other regulatory genes. This is called operon. Till now nothing is clear. This Lac Operon, it is an example of this operon. There are many such operons in case of prokaryotes, but in eukaryotes, polycistronic genes are not present. It is monocistronic gene. So in case of prokaryotes, if this is the transcription unit, then this 1, 2, 3, all these are structural gene 1, structural gene 2, structural gene 3, like that. And then it is controlled by a common promoter. It is controlled by a common promoter and many other regulatory genes are also there, which we are just going to see while studying Lac Operon. The promoter, terminator, everything will be there. This is in case of prokaryotes. This is in case of prokaryotes. This is polycistronic gene. Poly, many structural genes controlled by a single promoter. Come to eukaryotes. In case of eukaryotes, it is monocistronic. Means only one structural gene which is going to form one enzyme. It has every structural gene has its own promoter and own regulatory genes. This is the difference between the prokaryotic and eukaryotic genes. Lac Operon is one of the example of such operation of polycistronic gene. So how does this polycistronic gene work? That was shown by Jacob and Monod and we will study it with the example of Lac Operon. This is the structure of Lac Operon. Means the structure of the transcription unit. This is the transcription unit for that gene which is going to produce those enzymes which are going to digest the lactose. Remember that the bacteria E. coli, it does not prefer lactose. It prefers glucose. Glucose is its primary source of energy, easily available also, and it prefers glucose even if lactose is present in the media. Only when glucose is unavailable, for example, suppose this E. coli is in milk, then it will not get the glucose. At that time, it then takes lactose. And so to digest lactose, these enzymes are required. 3 enzymes. Just you have seen that the lactose, it is a polycistronic gene. But one important thing is that all these genes, the enzymes which they will produce, they are related in function. Means this Z, this Y, this A, these 3 genes, they will produce the 3 enzymes, all of which are necessary for the digestion of lactose. These structural genes, these three structural genes Z, Y and A. Z produces the enzyme beta-galactosidase. This beta-galactosidase, it is the primary enzyme which is required for this step, the breakdown of lactose into glucose and galactose. This Y gene, it produces the enzyme permease. What is the requirement of permease? This is E. coli and all around this is the milk. This permease, it permeates the lactose molecules in the environment to enter into the body of E. coli. So, it gives the permission to the lactose molecules to enter the body of E. coli. And then the A, it produces trans-acetylase whose function is not yet that clear. So, these are the structural genes and the enzymes produced by it. This one, this is the promoter. Promoter is that place, see, these structural genes, before forming these proteins, these enzymes, they will first form the mRNA. That means mRNA is going to form RNA, not DNA. So, the enzyme required will be RNA polymerase and not DNA polymerase. Okay. So, RNA polymerase is required, and this RNA polymerase, it is the P, promoter is the site where RNA polymerase attaches itself. So, this is the function of promoter. Now, come to this side, this is I or inhibitor gene. What is the role of inhibitor gene? Just now, we have seen that the bacteria, it prefers glucose and not lactose. Only when it is not getting glucose, at that time it is taking lactose. That means all the time it does not need these enzymes because it does not need to digest lactose. So, this gene, inhibitor gene, its role is to order that, to keep on hold the formation of these enzymes. So, this is inhibitor, this is promoter, which promotes and says to the RNA polymerase, okay, go on and transcribe these genes and then form the enzymes. Whereas inhibitor genes, it says that now lactose is not present. So, no need to synthesize these three genes. So, this inhibitor gene, its function is that it produces one repressor molecule. It produces one repressor molecule. What is the role of this repressor? This repressor, what it will do? It attaches itself to the operator. This is O for operator. It now attaches itself to the O position. So, what will happen? RNA polymerase is here. It cannot read the signals and it cannot move ahead to transcribe the Z, Y and A genes. So, this is the structure of Lac Operon, where you see the structural genes, its promoter, structural gene and promoter, this much is necessary to do the job. Then this I gene and operator gene, they are needed to stop the process, to keep this process on hold. And this P, what is this P? This P, this is the promoter for the I gene. This is not the promoter for these structural genes. This is the promoter for this inhibitor gene. That the inhibitor gene, which produces the mRNA, its RNA polymerase attaches to this site. So, this is the function of this promoter. This promoter is different from the promoter on this side, which is the promoter for the structural genes. Now, there are two conditions, that is Lac Operon in switch off condition, Lac Operon in switch on condition. Switch off when glucose is available and switch on when glucose is unavailable and lactose only is available. Let us see both these conditions. Let us study the structure of Lac Operon in switch off condition. When will the Lac Operon be switched off? When the glucose is available. So even if lactose is also available, but E. coli will be not taking lactose, it will only be taking glucose. So it does not need the product of these three genes, that is Z, Y, A. So what modus operandi is there? Let us see. The RNA polymerase for the transcription of this ZYA, it attaches itself to the promoter site. So what is this? This is RNA polymerase. Okay, but its work is not required because glucose is available. So this I gene or the inhibitor gene, what it will do? It forms one mRNA. This is its transcription. Then from this mRNA, it will translate to form the repressor protein. So it forms a repressor protein. So these two are its transcription and translation processes. Translation. Fine. So this repressor protein, what it does, promoter के सामने जाके वो बैठ जाता है. So it now moves to this place and it blocks the path of RNA polymerase. This repressor protein, it binds to the operator site and it blocks the path of RNA polymerase. So now it cannot see properly the start signals. So now these three genes, they are not transcribed. Not transcribed means mRNA is not formed. So naturally translation is also not there and the products are not formed. This is Lac Operon in switch off condition. Normally Lac Operon, it always remains in switch off condition because glucose is preferred by E. coli and glucose is so easily available. So rarely E. coli needs operation of Lac Operon. So it always keeps this transcription unit under negative control. इसे वो काम नहीं करने देता है क्योंकि उसकी ज़रूरत नहीं होती है. Now let us see the switch on condition when lactose is available and glucose is not. Now let us study the switch on condition of Lac Operon. Switch on. Why it is required to switch on the Lac Operon? That means the glucose is unavailable. Glucose is not available, but the lactose is available. So now E. coli switches to lactose. It will break down lactose into glucose and galactose and then he will use the glucose there. So here I have drawn the structure of Lac Operon. Let us switch on this one. First we start with RNA polymerase. RNA polymerase, it attaches to the promoter site. So this is RNA polymerase. Fine. The inhibitor gene, it will as it is produce a repressor gene. But that repressor gene, E. coli does not want to bind to the operator. But it is forming. So this RNA polymerase for this I gene, it will join to its promoter. This is the RNA polymerase for I gene. And then this I gene, it will be transcribed. So this is transcription of I gene to form one mRNA. Then this mRNA, it will be translated to form the repressor protein. So this is the repressor protein. This is the repressor protein. But now it will not go to bind to the operator. Why? Now it finds some other molecule for which it has more affinity than the operator. Who is that molecule? That molecule is lactose itself. Now lactose is available in the environment, and the little amount of permease which is present in the cell, it allows the lactose to enter into the body of the E. coli. So this lactose, this is lactose. Now these two will bind together. When these two they bind together, then this is the repressor protein to which this lactose binds, and thus this structure is inactive repressor. This is inactive repressor who is happy with lactose, and it will not go and bind to the operator. Done. So now this RNA polymerase, there is no patthar ka roda, there is no obstacle here in front of RNA polymerase. So it can move this way and it can transcribe all these three genes. These polycistronic genes, they are transcribed. When they are transcribed, then we get a common mRNA. So when this mRNA is translated, this mRNA is then translated and we get the three enzymes. And we get the three enzymes. These three enzymes are by Z gene, it is beta-galactosidase, which is the main enzyme for breaking up the lactose into two molecules. Then Y, which forms permease, which allows the lactose molecules to enter the body of E. coli. And the A gene, it forms trans-acetylase. Trans-acetylase. Done. So the whole switching on of Lac Operon was done by whom? Whom can we call the inducer of Lac Operon? Ki haan bhaiya, ki yes, due to this molecule, this molecule has switched on the Lac Operon. It is this lactose. So this lactose molecule, it is called the inducer. It is called the inducer. It is the one which has switched on the Lac Operon. It has withhold the repressor molecule, not allowed the repressor molecule to attach the operator. Thus, it allowed the Lac Operon to form the enzyme which is going to degrade it. That means our lactose has become the Bhishma Pitamaha who is telling how to destroy him. Thus, students, by studying the structure and working of Lac Operon, we have studied how the gene regulation is done in prokaryotes of a polycistronic gene. Isn't it? It was a polycistronic gene made up of the structural genes, more than one genes, the ZYA controlled by the same promoter and other regulatory genes. And in prokaryotes, this gene regulation means when it is not required, then it is switched off. When it is required, then it is switched on. This gene regulation in case of prokaryotes, it is done at transcriptional level. Rather, we write regulation of gene expression. The regulation of gene expression means whether it will be switched on or switched off In case of prokaryotes, it is mostly done at transcriptional level. You just studied transcriptional level. You just studied that, students, while studying the Lac Operon, कि उसका transcription ही बंद कर दिया जा रहा था. Whereas in case of eukaryotes, it can be done at various levels. What means from DNA, the RNA is not formed. So first is transcription level. Transcription level. Then the RNA which is formed, it is heterogeneous RNA. In eukaryotes, there has to be a post-transcription process to change this heterogeneous RNA into active mRNA. This is not done. Means at post-transcription level. Post-transcription level. Which process of post-transcription level is not done? It is the splicing. The capping and tailing is done. Splicing is not. Then when it is required, then immediately the introns are spliced and we get the active mRNA. We can stop it moving from nucleus to the cytoplasm. Means at the transport of mRNA from nucleus level. This also can be stopped in case of eukaryotes. And then it even moves into the cytoplasm. Then its translation can be stopped. So at translation level. So in case of eukaryotes, it can be at many levels. The gene regulation of gene expression can be done. Whereas in prokaryotes, it is at transcriptional level. One of its example, one interesting example you have studied, that is of Lac Operon. I hope that it was easy for you to understand. If the video was helpful, then do like and subscribe and share with your friends. Thank you for watching the video.